I hope that people understand what we are up against as it relates to trying to stay healthy, trying to stay alive when we're battling the ultra-processed industrial food complex. We've got a whole industry out there dedicated to feeding you good-tasting food that's going to kill you if you continue eating that. And when you come to this church, we are the opposite. We're trying to keep you alive. So we try to reinforce healthy lifestyle practices and ask you to mitigate what we all know is toxic, what is going to shorten our healthy lifespan, what is going to create disease states that wouldn't otherwise occur, and what's eventually going to create every major obstacle to achieving super longevity you can imagine. So a purpose of attending these monthly events, we hold 8 to 9 live services every year, is to remind yourselves what you already know, and that is there are very healthy ways that we can live, and there are very unhealthy ways. And one of the most dangerous habits so many people have gotten into are sedentary lifestyles. They don't move very much. Now, I showed this slide last month. It talked about walking, not a whole lot, but just enough to keep your body active and adding potentially 5 more years to your lifespan. This was a huge study published at the end of 2024, and literally 1 week after I presented this at the church, another study came out, and this study provided even more startling data. And for those people who are sedentary, which is getting to be the majority of Americans, they just get up and walk a few steps and sit back down again, your risk of dropping dead is 300% higher than if you do a little bit of walking. And if you do just a little bit of walking, you still have an 83% increased mortality risk compared to those who walk 10 to 15 minutes a day. That's not an awful lot of time, especially in South Florida, but you can go out and do it without freezing to death like the people of other parts of the country have to. And if you walk more than 15 minutes a day, you will extend your lifespan in a way that you won't die sooner by virtue of being sedentary. This is a huge problem, and study after study is validating that while we're going to talk about some aggressive exercise programs here tonight, even just walking 15 or more minutes every single day can benefit you in huge ways. And this study showed that people who are walking under 5 minutes a day, which really is trivial, they are at a much greater risk of suffering cardiovascular disease. And this does not necessarily mean you have to be very aggressive in your walking. Just remain active. Don't let sedentary lifestyles shorten your lifespan, because the data conclusively shows this is going to shorten your healthy lifespan. Now, walking is good, but there's also the high-intensity interval training. A lot of data has been published on that, and this is sometimes just 5 minutes a day of very vigorous, high-intensity exercise of some sort. It could mean climbing the stairs, walking very, very fast, whatever else you can do to get your body moving, as opposed to remaining sedentary, which frankly, I have to admit, and I always want to admit, because I don't want to sound like a hypocrite, I've written books that have sold millions and millions of copies, 1,500-page medical textbooks. Well, it took a lot of time to write those. For about 15 years, I sat at a desk for 15 hours a day, hardly moved. I was one of those sedentary people. I'm not that way now, but in my early career, I kind of had to be. So I always want to admit when I don't do what I was supposed to be doing, but now I am doing more walking, more stair climbing, more exercise than what I was doing before, and I'm in better shape now at age 71 than I was 24, 25 years ago, when walking just 6 flights of stairs would absolutely wear me out. But we've got conclusive data showing that high-intensity training, and that can be a combination of walking slow, walking fast, taking stairs, bicycling, whatever you can do, it pretty much benefits every aspect of your biology. You can correct major underlying health issues by simply being more active for just a brief period of time. Now, there's better ways to exercise. I mean, there's more that you can do than what I'm just showing you here. But you get all of these benefits from high-intensity interval training. Again, a lot of studies published on it, but it even benefits, of course, your mental health. So you get the health benefits across the board by doing something that's easy, something that doesn't really cost you any money at all if you can figure out how to do it the right way. So again, the power of reinforcement. Remind yourself that if you are sedentary, you are shortening your lifespan. It's a lethal habit that many, many people have fallen into. Now, we're going to talk today about the prospect of biological age reversal. And for a lot of people, they're afraid they won't live long enough to benefit from these breakthroughs that are occurring virtually every day in the research laboratories. But I always like to use historic examples to show how close we may be to finding a way to reverse aging in a meaningful way so you feel it, so your disease risks are back down to that of a healthy 25-year-old. And you look at the history of communications. You go back 3,000 years BC, they discover a form of paper so we could start writing things down and keeping a record of it. And then it took about, well, 4,000 more years to develop an efficient ink pen. But then you go to 1837 and the Morse code, an electrical pulse sent over a wire. That was the first means in which mankind was able to communicate instantly across long, long distances. And at just 137 years after that primitive Morse code was discovered, we have the iPhone 1. And what I'm trying to show you here, and this chart will make it a little bit more clear, it took 2,500 years to go from paper to developing recorded history, 5,500 BC. And of course, that's debatable because there wasn't much recorded back then. But it took a long time, 2,500 years, to discover paper, and then more time to discover ink pens and that sort of thing. But you look what happened, that 2,500 years to discover paper, and then 4,000 years for the Morse code to be developed. But once they developed the Morse code, only 137 years, and wow, we've got all of these technological advances occurring. So once we make a major discovery as it relates to what causes us to age and how we can circumvent it, then that technology can accelerate much, much quicker on an exponential basis. And that's what we're doing with Yamanaka transcription factors. These are genes that reprogram old cells to turn them back into younger cells that can theoretically regenerate our tissues forever. This was demonstrated in vitro for the first time in a petri dish, 2006. Well, for the first time ever, they were able to reverse aging in cells in a petri dish. Well, you move forward to 2022, 2024, they did it in live mice. They reversed aging in old mice using these Yamanaka transcription factors. And this year, we're going to start with monkeys. We're going to see if we can use the same technology that reversed aging in old mice and apply it to old monkeys. If those old monkeys grow biologically younger, our group will attempt to do some human projects in 2026. And the good news is many other groups are also doing this research, seeking a way to use these Yamanaka transcription factors to reverse aging. But if you're concerned you're a little bit too old, you may not be alive to benefit from it, I can tell you the pace of this research is accelerating so fast that within one to two more years, we may know so much more that we may be able to, well, grow people young again. This is what I'm looking forward to, and we're not alone. The wealthiest, the most influential, the most famous people in the world are all putting money into research aimed at slowing or reversing aging, finding better ways to treat disease, looking to improve the health of the human race. It's not only some of the far-out alternative health groups doing it. This is now mainstream. Age reversal has grown mainstream. It's being reported all over the place. And what we're hearing from people and what we're seeing in the media is that in 2026, it may not just be our research, there may be other people using different ways of inducing the OSK Yamanaka transcription factor expression in a way to reverse aging in people. It's moving ahead that fast, and we're seeing this occur. And we're having people even call us up for consultation. And we give that away for free. People want our information. We want to give them that information so maybe they will find that cure for aging. If they find it faster than us, I think everyone in this room will all be very happy. Whoever does it, it means we get to live a real long time. We don't have to suffer aging. Now, the Yamanaka transcription factors, the OCT4, the SOX2, KLF4, c-MYC, these are the original 4 Yamanaka factors. OSKM is the acronym. You'll see that talked about more and more as these studies come out showing OSKM, when it's introduced, when it's induced as it relates to expression, it enables old organisms to grow biologically younger. But the research we're doing and many others are just using 3 out of 4 of these Yamanaka factors. So our research is just OSK, which we believe will be both safe and not have that increased cancer risk. So OSK is what you're going to see probably more and more people talking about. Now, the first study that reversed aging in mice, the Salk Institute did it in 2022, and they did use the CMYC. They used all 4 Yamanaka factors, OSKM. They reversed aging in the old mice. There was no evidence of cancer, by the way, no side effects, and they were able to demonstrate something that had never been done before. An old mouse growing systemically, biologically younger. 2 years later, another group did another study using just OSK, and they were able to induce major reversals of pathological aging. The mice they used were the human equivalent of 77-year-old people. And they gave these old mice the OSK Yamanaka factors, and they were able to induce OSK partial reprogramming, improve frailty scores, and most important, I guess, extend remaining lifespan. If you're part of the control group, well, that's unfortunate. You died after 8.85 weeks, but the ones getting the OSK, they more than doubled their remaining lifespan. And that is absolutely spectacular to think if we're, let's say, a healthy 75 years of age, a really healthy 75, we're supposed to live to be around 88 to 90. If we can double that, wow, you get to live to be 115, 120. Think about that. If you get to live that old, well, at that stage, I'd like to think aging will be fully reversible. You might get to live indefinitely. So that research has already been done. What we're doing now is looking to improve that. We're looking to improve that because what's so fascinating is the scientists who did this study, they were only able to induce a tiny fraction of the intended OSK expression, and they still were able to achieve those remarkable systemic rejuvenation effects. So OSK is what we're seeking to add to your chromosomal DNA. This is the DNA inside your nuclear cell portion that controls cell functions. And there's a number of ways to do this. A lot of people will use viral vectors. They do work. They may have some side effects, so we're not using those in our research. What we are using are polymers. We're using plasmids. We're using lipid nanoparticles. These are ways of delivering OSK into your chromosomal DNA, and then once that OSK is in there, it induces the expression of pro-youth factors. It reprograms cells. And then chemical reprogramming, and I know that sounds hideous, but we're talking about small molecule reprogramming in which you'll take essentially some pills every day in a way that will cause the natural production of OSK in your cellular DNA. And that would be an ideal way of inducing this OSK. And that kind of research is being done by others, and it may be done in 2026. If it is, fantastic news. So I just want to explain to this group, I've shown this before, but it's important to understand how easy it is to add genes to your chromosomal DNA in order to achieve some potentially remarkable results. So let's just take the lipid nanoparticle as an example. It's a fat, a lipid, and we embed in that, and we're talking about nanotechnology here, the scientists embed the blueprint for the OSK into that lipid nanoparticle, which easily crosses the outer membrane of the cell. Absorbed into the cell, it releases the OSK mRNA blueprint, and that is then transformed by the ribosomes into OSK protein. And that protein then easily goes through the pores of the nucleus and binds to your nuclear DNA or chromosomal DNA. And when those OSK Yamanaka factors bind to DNA, they induce pro-youth signals. They tell the cell, stop growing old, start growing young. And as been demonstrated in vivo in mice, it works. And if we can demonstrate that in monkeys, and if others demonstrate it in people, we found a way, science being the people who do that, have found a way to reverse aging. So our 3 methods of delivering it, the research that we're doing, first, it is using OSK and mRNA. Another way to do that is a polymer with an OSK pDNA vector, a way of holding those genes inside a plasmid for delivery. And then there's that lipid nanoparticle that I showed you the example of. These are the 3 different delivery methods we are testing right now in mice to see which ones work better in monkeys. And as I'm going to show you, we're pretty much done with the mice. We're going to be moving on to the monkeys really, really fast. Now, this is a chart that I showed you last service, and I just want to make sure everyone understands what this represents. And what's so spectacular is this is the first time we used this particular method. We thought it would work, but we didn't know until we tried. So on the left of every one of those graphs is the control, and they have no OSK expression. They're old animals. This is, by the way, taken from live mice who had OSK injected in them just once or twice, a very low potency of the OSK pDNA. And you can see that there was expression of OSK demonstrated within 1 or 2 days. This is spectacular. We were so impressed with the results here. And this is another way that we've shown before. You can see the control mice, they didn't get any OSK, and they, of course, have no OSK expression. But by day 1 and day 2, we had robust biodistribution of OSK throughout the body of these older mice. And we're now testing those mice to see how young may they be growing. And we've already got preliminary evidence they are growing, well, they were slowing their rate of aging with just that one injection. Now, some people ask me what a plasmid is. It's a small circular DNA that is outside of the nuclear DNA, but it's used by scientists to embed genes in the plasmid. Plasmid then gets into the nuclear DNA in such a way that it releases the genes. So this is a way being used in the laboratory to deliver, to add beneficial genes to your chromosomal DNA so that you can benefit from what those genes induce. It's fantastic technology. So these are the benefits we saw with the P for plasmid DNA. We saw these nice increases in the multi-tissue samples of the OSK expression. And by the way, the bars to the very left, it looks like there's OSK, but the way the math was calculated, that is 0. To the very left, there was no OSK, and to the right are the improvements in OSK in response to the pDNA delivery. But then look at the mRNA OSK delivery. It's much more robust. Again, to the left of every one of those charts are the control animals. Those unfortunate animals normally aged. They don't have any OSK, but we were able to induce it by embedding the OSK into an mRNA molecule and having that systemically delivered to their nuclear DNA. So you see there are much more robust benefits with mRNA compared to the pDNA. And this is what really amazed us from this particular research. It was sustainable. The pDNA fell off after 2 or 3 days. We weren't seeing the OSK expression very much, but the OSK bound with the mRNA, it was able to produce sustainable OSK expression up to 21 days or longer. We stopped testing after 21 days. So we were able to demonstrate that potentially within the next year or two, you'll have a syringe delivered to your door every month. You'll shoot up your mRNA OSK and boost your OSK expression, hopefully grow biologically younger every time you do it. That's our objective with this kind of research. But the idea that we're getting sustained OSK expression is a very, very, very good sign. So to give you an idea as to how fast this is moving, and I've been involved in research for almost 50 years, almost everything goes very slow. This is fast-tracked for all kinds of reasons. Some of our supporters, they're in their 80s or 90s, they need this or they're going to die. But if you look, July 2025, I presented this chart at RADFest, and you can see we've completed some aspects of the study. But by last month, on October, this is what I showed you, we were almost done. We were just waiting for the lipid nanoparticle work to be done on the mice. And if you look at where we are now, November 2025, we've completed everything. We're now ready to test it in primates. So this is something that I'm going to announce in just a few minutes. And by the way, what you're going to see right now has never been shown to a public group before. These are unprecedented research findings. They were revealed to me November 19th. I had a long night last night putting this together to make sure people understood we've made substantial progress over the last 30 days alone. And this is using lipid nanoparticles to deliver the OSK into our nuclear DNA. Remember, the 4 Yamanaka factors, the OSK, this is what we're seeking to put into your cellular DNA so that you get to grow biologically younger. And again, the different methods of doing it, viral vector, polymers, lipid nanoparticles, chemical reprogramming. We're studying right now the lipid nanoparticle to compare it with the polymer mRNA to see which one works better. We intend to test both, by the way, in the monkey model, and we're also testing them in the mice. So here are the preliminary results, which again, unprecedented, and we didn't expect to have success this early on. The very first time we put our lipid nanoparticle OSK construct into these old mice, we were able to see very rapid, consistent increases in OSK transfection or delivery. So you look at the control to the left, they don't have any. They didn't get any of the LNP, lipid nanoparticle OSK. But you see in the heart and the liver huge increases in OSK transcription and, we believe, expression. Now, next month, you're going to see many, many other organ systems. These are the first 2 samples we received back, and the scientists who did this work for us, they said, we've never seen this happen so quickly. We thought you'd take 10 or 20 tries before we ever achieved this. We achieved it the very first time with the lipid nanoparticles, the very first time with the plasma DNA, and the very first time with the mRNA. So we're hitting right now 1,000 as it relates to what we're seeking to accomplish before we go into the monkeys. So what you're seeing here, by the way, I want to make sure this is clear. This is the transfection or delivery of the OCT4 Yamanaka factor into the heart and liver. This is the SOX2 transfection into the heart and the liver, and this is the KLF4 into the heart and liver. And again, there's multiple other organ systems that we're getting data back on every day from the lab as we send them tissue samples and they analyze them and get the results back to us. So we're achieving systemic induction of OSK in old mice that we hope to induce into the old monkeys. And this is a summary by the lead scientist as to what we achieved here. That word transfection, it means delivery of the OSK into the DNA of these mice, and they tested separately O and S and K. To think that nanotechnology has advanced that far where they can not only produce it and deliver it, they can measure it. They can measure how much is getting into the nuclear DNA, and we're also going to be able to measure what type of age delay, age reversal effects are occurring. So what we're seeing here, and look at this, November 17th, this is the report that I was given. This is how far we are along. We are literally on the front lines right now of a major, major scientific advance. And we did safety studies on these mice. We didn't want to kill any monkeys by giving them too much, so we gave varying doses. And the good news is we were able to ascertain what may be the ideal dose for the African green monkeys that won't create any toxicity, but may induce significant age reversal if it works as well in the monkeys as it does in the mice. So again, this is very current data. When you come to this church, you learn information before the rest of the world does in certain cases, certainly with this research. So at this point, we're taking the polymer pDNA and a polymer RNA, and we're testing that to see how well it may have induced age reversal effects. And then we will, of course, move into the monkeys. So this gives you some preliminary data as to which one is winning the race. Now, you look at that green check by these different boxes, that indicates a slowing of aging. And if you look at the mRNA column, that's to the very far right, we're checking all the boxes. We're only missing one there, whereas the pDNA only checked one of the boxes. This is a preliminary report, but at this stage, it looks like the OSK RNA construct and the OSK lipid nanoparticle construct, they're working about the same, and we're going to test both of those in the African brain monkeys. And the announcement I'm making right now is December 9th through December 12th, we will be injecting old monkeys with the mRNA and probably the lipid nanoparticle OSK. We'll be looking to see what effect it has. We're doing aggressive baseline measures, aggressive interim measures, follow-up measures. We want to ascertain, number one, are we transfecting the DNA of these older monkeys, and are they growing younger? There's all kinds of ways of measuring that, and by next month's service, I'll be able to give you a little bit of update of what's happening with that particular research. And you're the first group. You're the first group in the world to really see this. There's people who pay hundreds, not thousands of dollars to attend scientific conferences, good science presented. I don't think anything like this has ever been presented at a scientific conference that I've attended. There's just, it's still in its infancy stages. What we know today, well, it's good. We're going to know a lot more a month from now, 3 months from now, 4 months from now. So we're going to, I'm going to reinforce right now the fact that people are not always behaving the way they should. I think a lot of people at this church do, but most people don't. We're engaging in unhealthy practices, and we're losing very good people I've known for decades because they're doing what they shouldn't be doing, and they know it, but they nevertheless deanimate anyway. So most of you may have heard of Peter Attia, a brilliant medical doctor. He wrote a book called Outlive, sold over 3 million copies, New York Times bestseller. And if you read the book, you'll be very familiar with a lot of what he talks about, because most of you know a lot about what he talks about. And the good news is he got on CBS News 60 Minutes a couple weeks ago and gave a very good interview about extending average lifespan. Now, he's not an immortalist, and I want to emphasize we do not agree with everything he says, especially as it relates to people over 75, because we have some people in this audience right now who are way over 75 and they're in really good shape. I'm going to have them stand up maybe after this little interview. Dr. Attia's book talks a lot about preventing cardiovascular disease, and he brings up something that we've talked about for many, many decades, and that's an LDL particle called apolipoprotein B. And when you have high levels of apolipoprotein B, unfortunately, your risk of heart attack and strokes, it goes up quite a bit. This is a measure of the atherogenic particles in your blood, and you want to keep it at a very low level. Now, last month, I talked about also C-reactive protein and the risk that poses to cardiovascular disease if the levels are elevated. Women with just slightly elevated C-reactive protein, they increase the risk of a stroke 69%, 74% increase in cardiovascular disorders and coronary artery disease. I mean, it's a deadly syndrome, or it's at least inconvenient. You wind up in a hospital being stented or open heart surgery. You don't necessarily want that, but if you have high CRP, you're a woman, you have over a 200% increased risk of coronary artery disease. So you want to keep your ApoB low, your CRP low. You want to do that as much as you can. And what they did in this study is they combined the evaluation of C-reactive protein and apolipoprotein B, and they found that it was a very effective way to measure one's future risk of cardiovascular disease. And what Dr. Attia advocates, and what we've advocated decades ago, is keep your C-reactive protein and ApoB levels as low as you're able to get them. Because if you're able to do that, you probably will reduce your risk of a cardiovascular disorder. And this particular study showed just one test of ApoB and CRP, it's a very strong predictor of your future risk of cardiovascular disease. And if your risk of cardiovascular disease is elevated, well, do something to lower it so you don't have that heart attack or stroke when you're supposed to. All kind of ways of making that happen. And this is something that I have talked about ever since I founded the various organizations I do that advocate healthy lifestyle, and that is defy conventional reference ranges. Because this is what you'll see if you get an apolipoprotein B blood test. You'll see that ideally you want to be under 70, but up to 100 is okay. You're low, normal risk. Of course, cardiovascular disease, #1 cause of mortality and morbidity, that you don't want to be normal. So the standard reference ranges are under 70, which is fine, but it's not nearly low enough. Look at what the data really reveals. Anything over 11 to 25 increases your risk of having a heart attack or a stroke. And every couple elevations that you see there further increases. You can see massive increases when it goes up to 40, up to 90, 100. And yet conventional medicine, if they see at 95, they say you're pretty good. And the reason they say that is they're used to seeing people over 120, 130. And I've looked at hundreds of various blood tests from people who think they're doing everything right. They often have very high apolipoprotein B levels, and I tell them you got to get that down. And frankly, the way to get it down is statin drugs, PCSK9 inhibitors like Leqvio, Repatha, ezetimibe. There are drugs out there that lower lipids, and they also lower apolipoprotein B. So if you're elevated, consider those drugs that alternative medicine in the early years attacked, and that's because some of them killed people, like Baycol. That was a statin drug, never should have been approved, killed people, it was withdrawn. But the current statin drugs don't induce those kind of problems. But I want you to look at that chart and understand that while conventional medicine says just get your ApoB levels under 70 milligrams per deciliter, you really want to get them down a lot lower. You want to get them down a lot lower. Now, Dr. Attia, he wants you in the range of 30 to 40 milligrams, and I totally agree. You want to get down to that very, very low range. He emphasizes as low as possible and as early as possible. And see, looking at the people in this room, there's a few younger people, and you've got a real advantage over us. You can get a blood test at your doctor's office, wherever, and see where your levels are and knock them down at a younger age. Or older people, we can lower them, but we're not going to benefit as much as younger people who lower them at a younger age. Dr. Attia does not want to see levels above 60 in his patients, and we certainly agree. You want them as low as possible. These are my blood test results. And again, I've looked at hundreds of different results, and these are the lowest I've ever seen. And I've had to work at it. This didn't come naturally, but I got my apolipoprotein B levels down to 24, and my C-reactive protein down to a virtual non-detectable level. And that involves a lot of prescription drugs, a lot of nutrients, a lot of dietary modifications. It takes a lot to get these kind of results. But when you do get a blood test result from your doctor, and or let's say a blood test, and he's prescribing a standard CBC chemistry, ask him to add ApoB and CRP to the test. And that could give you a real nice picture of what your real cardiovascular disease risks may be, and gives you a target to aim for to try to lower it, to try to get it below what the conventional reference ranges show you. Because I've advocated for my whole career, defy what conventional medicine tells you. Conventional medicine has always erred to the high side because normal people are high with LDL, high with blood pressure, and since doctors see so much of it, they consider it normal. You want to keep your blood pressure low normal. You want to keep your lipids, your glucose low normal. That's how you get to live longer. So again, this chart showing incremental increases in ApoB causing massive increases in cardiovascular risk. You want to keep your ApoB levels low. It's part of the reinforcement that we try to do at this church to emphasize what you can do to live longer. Again, my levels, I achieved them. It took me several years to make this happen, but I worked at it. I didn't just let nature take its course or make a few minor changes. I underwent major, major changes to enable those kind of results to occur. And again, to conclude every presentation, please, if you think you can avoid a problem by not having it diagnosed, or if you have something diagnosed, pretend like it's not there. It just doesn't work. It doesn't work. I'm a professional at it. I've talked to people for decades who have issues. Those who correct their issues continue living. Those who allow their problem to fester in their body, it worsens, and they die prematurely. And they pretty much knew they were dying prematurely by virtue of their unhealthy lifestyles. Many were unable to change it. So I appreciate the big turnout this month, and I'm here to answer any questions people may have.